Hey again, everybody. Time for a new coffee cup. I hope you are getting used to these videos. Sometimes I think it's very difficult. I'm trying to keep them as short as I possibly can so that you can get the information, tag along, remembering all the different videos. Some I hope are fun that I'm posting. And of course, you can keep up with the readings in the textbook as well as the more detailed slide sets that are available in Piazza. So today we're going to tidy up. We're going to talk about these guys right there. They are not jellyfish, given we're so close to the ocean, but rather these are ribosomes. And of course, they are the motivating factor behind translation. So we're following up on our ideas about central dogma and getting ready to talk about the bottom portion of the chart, talking about translation. You've already seen ribosomes. We're going to talk about transfer RNAs and the construction and continuous transfer of the information from messenger RNA to a polypeptide and or protein. Of course, what we're really talking about now is the complex right-hand box of the side that occurs in the cytoplasm. And so, say I can poke over here and see you, and I can even point from this direction in the right-hand portion of the side and talk a little bit about what happens in order to get things moving from the messenger RNA into these novel products. And you can see it's a fairly complex process. And over the years, we've really added in a lot more information than we had in the beginning. Now, I'm going to try to keep this to the point where you get the information that you need, especially when we start talking about some of the more technical aspects of genomic sequencing, personal genomics, and some of the techniques that are very commonly used in genetics and molecular biology these days, which will really start next week. But I don't want to go into overwhelming detail. Okay, that's really for a later course in molecular biology and to some degree after you've had biochemistry. So we need to know a little bit about what in the world is a protein. And of course, we know that proteins are made up of an amino acid. Okay, proteins, of course, being one of the macromolecules of life, and not only one of the macromolecules, but it is a polymer. That is, it is a string of smaller subunits, and of course these subunits are amino acids. Now, if you don't recall some of this information, I'm going to go over it pretty quickly. These are ideas and subject matter that you should remember from Bio 1. And so we're just going to go over them kind of cursorily. Please remember there's an excellent review in the textbook. Check your Bio 1 textbook. And of course the slide sets. So here we see the typical view of an amino acid. An amino acid is a central carbon with an amino group. Here's that NH2, an amino group on one side, and a carboxy group, which of course is an organic acid, C double bond O, OH. Okay, so we see this is the carboxy group, an organic acid, hence amino acid. Each amino acid has this basic structure, but also has a side group, sometimes called a residual group, a side group on the molecule, which differentiates it. And I've got a slide here showing you all 20 of the amino acids. Again, this is something that you should have seen in Bio 1. You certainly should remember. And all of the amino acids are classified into several different groups based on chemical properties that are given to the molecule as a result of the type of side chain. And I like this picture because it shows you in the green boxes the basic amino carbon acid structure, or the amino carbon carboxy structure of the amino acid. And then, of course, underlying the green box are all of the different possible side chains. These side chains give different qualities. They can be hydrophobic or nonpolar, hydrophilic or polar. They can be acidic or basic. Many of them are branch chains, some of them are single chains, and it really is these side groups or these residual groups that dictate how the amino acids interact within the protein itself. Now, there are 20 of them. I usually tell students, if you look at the names, you should certainly be familiar with the names. Glycine, alanine, valine, just reading the three off of the top row. And notice underneath each name is the three-letter code designation for each of the amino acids. In most cases, the first three letters are just the first three letters of the amino acid itself. However, you could see if you wander down to tryptophan at the end of the top row, tryptophan is TRY, whereas its three-letter abbreviation is TRP. Notice asparagine and glutamine are a little bit different. Asparagine in the second row under the hydrophilic or polar side groups. Asparagine is ASN, not ASP. The reason for that is ASP is aspartate, and which is down the first one in the third row of amino acids. You should be familiar and be able to figure out what the amino acid code is from the three-letter code, and you'll see we use these very commonly. I will tell you right now, it is an excellent idea to learn the single-letter code, which is the third abbreviation in brackets underneath the name of each amino acid in each row. They are mostly easy to figure out. However, there are a few tricky ones. I am not going to require you to memorize them at this point, although I may ask you at some point in time to use a codon usage table and utilize the single letter code just to get you in the habit of using them. Will I actually ask you, for example, what is the amino acid represented by a Y? Well, you don't have to memorize that it's tyrosine, but you'll see if we have the codon usage table a little bit later, they usually have, and the ones I'll give you will have the single letter code. It's incredibly important to have these abbreviations and to learn them, because when you're dealing with genes that could have hundreds of amino acids, you just can't write them all out in a sequence. And even using three-letter codes for the amino acids, it could take up many, many pages of text. And therefore, it's the single-letter code that have become prevalent in nearly all of genetics and biochemistry at this point in time. So take an opportunity, read these tables, spend some time looking them over. Wait till you get to biochemistry. I know in biochemistry you will actually have to memorize the structure of all the R groups. Although I understand Dr. Keenan doesn't require you to know the single letter code in biochemistry, but I, on the other hand, will require you to know the single letter code if you take bioinformatics with me, and I'm sure Dr. Luke also requires it as well. When we think about exactly how peptide bonds are formed, you can see on the left-hand side we've got two amino acids. The R groups, of course, are unspecified, just two different amino acids. And notice if the carboxyl group and the amino group, and we remove water, we make the characteristic peptide bond, which is C double bond O N C, and you can see kind of this peptide bond. It is made by a chemical reaction. This is called a condensation reaction because we remove water. Okay, water is a byproduct. This is done enzymatically in the cell and happens very, very quickly. The peptide bond is one of the strongest chemical bonds in all of organic chemistry. Just to give you some idea of how tough it is, if you were to digest the peptide bond and try to do it chemically in the laboratory, you would have to do it using concentrated hydrochloric acid under boiling conditions for about 12 hours to completely break apart the peptide bond in a given protein into its individual amino acids. So that is a pretty tough bond to break. So peptides and proteins actually are very, very strong. Think in terms of spider silk, think in terms of your fingernails, think in terms of even your hair. It's very tough. Of course, the amino acid sequence that's dictated by the DNA and therefore the messenger RNA only indicates the primary structure or the primary sequence of the peptide or protein. And so you can see here's a 4 amino acid peptide. Some people ask what's the difference between a peptide and a protein. There is no strict definition other than peptides tend to be smaller, polypeptides tend to be intermediate in size, and of course a protein is a full-blown, large-scale polymer that has advanced qualities. Now we'll see, because of the interaction amongst and between the R groups, proteins tend to form several different types of structures. There is then the secondary structure, which is the three-dimensional way that the amino acids begin to form and merge together. In this particular example, we see an alpha helix. Of course, there is not only an alpha helix, there is a beta sheet. So the secondary structure of the molecule can be in helical form or in sheet form. Again, completely dependent on the interaction of the R groups in the primary sequence. In addition, all of the different secondary structures can interact to create a larger three-dimensional structure called the tertiary structure. And if we put together multiple polypeptides, again, all of those peptides interact based on the properties of the primary and secondary sequence. It creates a quaternary structure. So primary, secondary, tertiary, quaternary. These are 4, also numbered 1, 2, 3, 4. And of course, the classic example of this is hemoglobin, which is the result of a 4-mer, meaning there are 4 molecules, and they are in particular a pair of alpha globin and a pair of beta globin polypeptides that merge together to form the intact protein molecule. And so you could see that's how this indicates here in a nice three-dimensional structure. I like this slide because periodically students want to know exactly what it is about those R groups that cause them to interact, and there are a handful of different types of interactions that occur. We have, of course, our familiar hydrogen bond that can occur among and between R groups within a peptide. There are ionic bonds, again, between the acidic and basic amino acid side groups. There can be hydrophobic interactions, and here we see 2 of the same R groups. When they come in contact, they actually push each other away, so they're hydrophobic. And a very, very common one, of course, is the disulfide bridge. The disulfide bridge occurs when two cysteine amino acid residues can form this disulfide here. This is a really, really important one and does quite a bit to define the three-dimensional structure of the molecule. There are also some weaker ones, for example, van der Waals interactions, and these are among and between these usually hydroxyl residues between the R groups of adjacent amino acids. So these are the most important ones for you to remember at this point in time are just that hydrogen bonds, disulfide bonds, and of course ionic bonds. Notably, again, please review the information on amino acids and peptides if you don't recall or if you feel that this is new to you. Ultimately, what we really want to talk about here are ribosomes, and we've got two different graphs here, one being, of course, the prokaryotic ribosome and the eukaryotic ribosome. In this case, we're using the mammalian ribosome as an example. Note that the prokaryotic ribosome has 31 proteins in the 50S subunit. Okay, and the S is for Svedberg unit. It is a unit that defines how far a molecule or a set of molecules will travel during density gradient centrifugation. And so the further it travels, the heavier it is. So we've got a 50S unit and a 30S unit in the prokaryotic ribosome. These subunits are made up of proteins, ribosomal RNA, and a couple of different types of ribosomal RNA. They bind together when they are assembled completely, and you'll see the assembly in a couple of videos shortly, which will come after this one, and you can see it creates an overall size of about a 70S ribosome. So the 50 and the 30. Note they don't add up. Okay, they never add up because this is all about how far these molecules travel within a dense, usually sucrose, sometimes salt solution. Now notice the complexity of the eukaryotic ribosome. Here we see that there are a much larger number of proteins in the eukaryotic ribosome as compared to the prokaryote, multiple types of RNA, and there are a 60 and a 40S subunit. What do you need to remember? Prokaryote is a 50 and a 30S subunit. Eukaryotic is a 60 and a 40. Obviously, 60 and 40 are bigger than 50 and 30. So you need to remember that. And of course, the functional unit is 80S on its own. The general manner in which translation occurs, again, transcription seems relatively simple compared to translation. And notice that we've got the ribosomal subunits in conjunction with messenger RNA. Okay, and we can see that multiple ribosomal subunits can line up on the same mRNA molecule, and each of them undergoing the elongation step of forming a peptide. There are different things that control the elongation step. There are different factors, different enzymes. I wouldn't worry about it. And each of these will create a nascent peptide. Nascent meaning newly born or brand new. So really complicated, but I want you to see the process, but mostly understand the basics. The synthesis and processing of the 30S unit really comes from a single ribosomal RNA gene in E. coli. It is cleaved into multiple portions by endoribonucleases. Of course, the ACE tells you it's an enzyme. Endo means it's inside the ribonucleotide. So it's cutting within the RNA sequence and then ultimately cleaved again by more endonucleases and exonucleases as well. Exonucleases trim from the ends inward. Just to give you an idea and take a look at how much more complex RNA processing is in the mammalian species, you can see here that there are a variety of these degraded regions, almost as if they are intron-like, to create the variety of different size of the rRNA species, which eventually become assembled into the ribosome itself. So, just a little background, there are 7 rRNA genes in E. coli. They are located in 3 different positions along the chromosome itself. However, in eukaryotes, and again, remember the size of the genome when comparing eukaryotes to E. coli. There are hundreds to thousands of copies depending on the eukaryotic species. They are always present in tandem arrays in the nuclear organizing regions of the chromosome, and the 5S ribosomal RNA genes are actually distributed over multiple chromosomes. So we really have a very, very different organizational structure between the prokaryotic and eukaryotic ribosomes. And that really brings us to the end of talking about translation. This is the functional way that ribosomes work inside your cell, the mechanism that they have. And we're going to use a separate lecture here to talk about the genetic code, transfer RNAs. So this is really will be one last step, and of course when we hit that end, which will be up shortly, available to you, that will be the end of the content that will be on the next exam. Hope you guys are staying safe, taking care. See you on the next filming.